Awesome. Okay.
Cool. Good.
So feel free, all of you, to pause me and to ask questions, because I like to talk. I mean, please feel free to just chime in with questions. No protocol. So let's make it as a conversation as much as we can. And I have the privilege of only being here with you, so we have a lot of leeway to do things in the way that we want. So I like to start by saying that I like to call myself a quantum engineer. This means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit surprised when I tell them that I study things that might be going on inside birds, inside butterflies. So before I explain where I'm going, I really need to tell you where I come from. So we're going to start by talking about hardcore quantum mechanics, and by the end of the talk, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, to radiation, and I hope to have convinced you that I think we can learn with nature how to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So humankind, I believe, is obsessed with measuring things better, because this might mean, and actually I think it means, that we understand nature better. So for example, we can think about measuring better frequencies. This is an atomic clock that sits in the National Institute of Standards and Technologies, the NIST, in the US, and it measures very well what a hertz or what a second is based on atomic transitions. One could think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. And we could, for example, think about measuring better acceleration so that your gaming experience is enhanced. Those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. There's actually a whole body of mathematical arguments to prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. So in my past, I worked with such a technological quantum sensor. It was like a quantum enhanced technological sensor. And I'm going to start my talk by describing it a little bit. And the reason why I need to talk about that is that I will then show you many analogies between this technological quantum sensor and quantum sensors that can actually be found in nature. So that's where we're going. But that's my past. I started my career putting a single spin in diamond to work as a magnetic sensor. So spin, right? Some of you might not remember what a spin is. It was, maybe you've only seen it in high school chemistry. Spin is a fundamental property of matter that is as fundamental as charge and mass. And electrons have spin, atomic nuclei have spins, photons have spins, even if it's zero. And spins, the quantum property of spin, it doesn't really have a classical equivalent, and it measures how well a quantum object interacts
Clarice,
with
can I interrupt for a
field.
second?
Yes.
Maybe for those who are not physicists, you can explain what the Stern-Gallach experiment is, because that's how the spin was found first time, no?
Sure. Cool. Okay.
Just quickly,
So,
because that's kind of the first manifestation.
So what was it? Was it in silver?
I think, yeah, exactly. Silver atoms. It was a beam of silver atoms through a magnetic, it must be a non-homogenic
gradient.
And then and then it splits up in two, no? which you can't explain classically, no?
In 2 or 3. I thought silver was 3.
I think it's 1/2.
But okay. So, but let's make it with electrons. The idea is that if you have a beam of electrons and you shine it through a magnetic field gradient, right? Like if spin didn't exist, or if electrons had spin 0, if you had like a screen that shows you where the electrons hit, if the quantum object that you're shining through this magnetic field gradient has spin 0, you're going to see one tiny spot at the screen. However, when people started doing this sort of experiment with different atoms or with beams of electrons, for example, with a beam of electrons, if you shine then through a magnetic field gradient, it turns out that after the magnetic field gradient, the beam splits in two, and you see 2 different spots at the screen. The reason why that is because electrons with different spins interact differently with this magnetic field. That is, one type, the electrons with one type of spin are deflected upwards. The electrons with another type of spin are deflected downwards, so that you have an effective splitting of this beam. And this is super weird. And I think that's, as Gerhard mentioned, that's how people actually, was it predicted before? I don't know.
I forgot now. I forgot. But I remember that Gallach experiment is the first experiment, I think, where it manifested itself. If it was predicted or not, I don't know. No, I'm not a historian.
Isn't it part of Dirac's relativistic electrodynamics?
If you will, yes.
Yeah, but Dirac is probably later, I think.
Later than Stern and Gallach, really? Okay. I mean, I don't know.
Yeah, Stern Gallach was, I'm just checking here, 1921. And I think Dirac was later.
It's probably later, yeah, a bit later. But yeah, it's in those years, in those very intense years for quantum mechanics, right? And actually, up to this day, people are using Stern Gallach like setups to do,
of course, yeah,
the spins of electrons and of other quantum objects. But physicists and chemists and engineers usually represent spin with an arrow. And I bring my props. I'll tell you if I want you to look at my props. So arrow up or arrow down represent just 2 different energy levels of this quantum property called spin. If you will, if you shine a beam of electrons through this magnetic field gradient and the beam of electrons is split, the electrons in this part of the beam have a certain spin energy, the electrons in the other part of the beam have another spin energy, right? So the first thing that I want to talk to you about is how I put a single electronic spin in diamond to work as a magnetic field sensor. Okay, so here's the promise, and I'm going to show you how that works. So this particular spin that I worked with, it arises from a lattice defect in diamond. All crystalline materials have different defects in the solid, right? And the particular defect that hosts this electronic spin is a so-called color center. A color center is a naturally occurring crystalline defect that is so called because when you shine light at it, it gets excited, it absorbs this light, and then it de-excites by emitting fluorescence. And color centers are the crystalline defects responsible for the color of those nice diamonds that you see there at the bottom of the slide. The particular defect that I worked with is a defect that is very hyped today. It's called the nitrogen vacancy center. it occurs naturally, but it can also be engineered. And it takes place when a nitrogen, which is the most commonly occurring substitutional impurity in the carbon lattice, in the diamond lattice, sits adjacent to a vacancy, to a missing carbon atom. And when those two things come together, there's like a mess of electrons that interact, right? There are the electrons close to the vacancy that are unpaired. The nitrogen brings one extra electron. There might be like charges coming from nearby to this defect. So it's sort of this electronic mess, but something almost magic occurs when you start to calculate the allowed quantum mechanical energy levels of this electron mess. And it turns out that the allowed quantum mechanical energy levels of this compound really look like the quantum mechanical energy levels of a single electronic spin. So what you do is that you can forget about all the things interacting, and you can treat this nanoscopic atomic defect as an effective single electronic spin. Right? And the way that we find this in the lab is you take your diamond slab, and it can be a very crappy diamond slab. The very first diamond that we worked with was like a jeweler discard or something. So you take your piece of diamond, you put it on top of a moving stage, and you sort of scan it around at the same time that you shine laser of an appropriate energy on top of this moving diamond. Now, it doesn't need to be a laser. Color centers absorb energy, get excited, and fluoresce even at room light, ambient light, as you saw with the picture of those diamonds in my previous slide. But we do this with the laser in the lab because it's more efficient and simpler. But the idea is that as we're scanning this diamond, the moment that the laser hits one of those naturally occurring defects, we expect this defect to absorb the light, get excited, and then relax by emitting fluorescence. And this fluorescence we can actually collect. And this is what it looks like at increasing zooms. So to the very bottom right there, what you see is a blob. And it's quite impressive because this blob is actually the fluorescence signature of one single electronic spin. The defect itself is nanoscopic, is very, very tiny, but you see a 500 nanometer blob in diameter because this is a diffraction limited image. But nevertheless, it's really cool because just by looking at fluorescence signals, we can actually find and if we know how to play with single electronic spins in diamond, everything at room temperature.
Clarice?
So yes.
Sorry, a couple of questions. So this effectively sort of single spin that emerges, is this coming from the a correlated effect of the electrons interacting with each other? Is this how does this emerge? It seems magical somehow that given the fact that it's atomically very complicated, nitrogens and carbons and lots of things floating around, what's the basis? Is this
and then there is also a dipole involved because it absorbs light, gets excited. So I actually do not have a better explanation to that. I don't think it's like a correlated effect, except for the fact that there can be a electron that can sit there with those very simple energy levels.
Oh, I see.
Jeremy, the similar effects are seen for doping in semiconductors. You know, I worked quite a lot on that. I did some femtosecond experiments on that. And when you have doping in semiconductors, you know, semiconductors are very well studied because of the semiconductor industry. Like doping in silicon is like the basis of everything we do, nah? More or less. And the thing is, it depends very much on the density. So if the density is low enough, then the defects are separated. And on average, now there might be two which are close together, then they act like H2 molecules. But if the density is low enough, most of them will be separated, and each one of the defects acts like an atom, except that there's a dielectric constant, there's a polarization of the whole. There's a dielectric constant in the diamond, so it's you have to take account the screening for the properties of this atom, yeah. But if they are separated from each other, they act like atoms, yeah. But if you would have a huge number of these defects so that they overlap, then you will get bands. Then they will overlap. If there's a relatively low density, you'll get like molecules, and if you go higher, you'll get bands. Yeah. So, but they are like single atoms. Yeah.
And as Gerhard was saying, is like you can think of it as a single atom, a single two-level system, or a single discrete number of levels within the band gap of diamond.
Exactly. Yeah. Of course, yeah, they will have different, this defect will have different levels, yeah. But, the lowest level depends on the defect, depends what type of defect it is. For some defects, you'll have, it depends on something like in chemistry, like electronegativity, no? But it depends what type of defect you have. But for some defects, you'll have levels inside the band gap. So that is then totally localized. But the higher excited states, of course, will be in the conduction band higher up, yeah? And they will be spread out. No?
So the color that the individual defect fluoresces at, is that characteristic of the diamond or characteristic of the defect?
It's a characteristic of this defect. The zero phonon line, that is the minimum energy that it needs in order to be excited is around 637.
Hello?
I'm thinking, I said the minimum or maximum? The zero phonon line is the minimum wavelength, right, of energy. The zero phonon line is at 637 nanometers, but of course it's much broader than this. At room temperature, you have an emission spectrum that is super broad.
Mm-hmm.
Right.
Jeremy, it depends on both. It depends on the crystal and the defect, no? Both, of course. So if you put the same nitrogen somewhere else, if you put nitrogen into gallium arsenide or silicon, you'll get a completely different spectrum.
And there are other defects in diamond, for example, that have zero phonon lines in different places and so forth, right? But here we don't care right now, and I'll tell you a little bit why. We don't care exactly. Other people care, but I don't care at exactly what wavelength this diamond defect is emitting. Okay, I'm actually only going to care about like how much overall it's emitting, because this defect has something that is even more magical, and it has to do with the existence of metastable states, and I can explain if you want. But this defect presents something that's called a quantum state dependent fluorescence intensity. This means that just by looking at how strongly this blob is emitting light, I can actually infer if the spin is, say, up or down, which spin energy state the defect had. In the quantum field, this is referred to as state dependence quantum readout, because just by looking at the fluorescence level, you can actually read out the quantum state. And that is super important for you to know what's happening with your system. So apart from the fact that it's super convenient that if we have fluorescence, we know that there's a defect, and you know that it's a single one, right? And then just by looking at mean intensities, you can actually have a quantum measurement. So this is super unusual in general. But that's why people started looking at this defect as a technological quantum sensor, and in particular, as a very good magnetometer. And here's how this works for it to be like a magnetometer. I depict there two of the spin quantum states that I call 0 and 1, which would correspond to, say, arrow up and arrow down. The difference in energy between those two states is in the microwave, is about 3 gigahertz. And it turns out that one of those states, the state that I call 0, is insensitive to the presence of magnetic fields. Okay? That is, if the diamond containing this defect is immersed into a ambient magnetic field, level 0 won't care. Whereas the level that I called +1 is sensitive to magnetic fields. That is, in the presence of an ambient magnetic field, that +1 energy level will be shifted a little bit by a quantity that I call delta here. And of course, delta by something that is known as a Siemens interaction. This delta is proportional to the magnetic field absolute value, right? So, and the next point that I'm going to make, it's actually, it what underlies all of quantum sensing. And this point is not very usually made. The problem of measuring a magnetic field is thus mapped onto the problem of the experimenter knowing how to measure a detuning, a shift delta from a known resonance, from a known energy difference. That is the energy difference between 0 and 1 in the absence of magnetic fields. Again, that's how all quantum sensing, if not all, like 90%, 95% of all modalities of quantum sensing works. You have a certain resonance, a certain energy difference that is sensitive to a certain parameter, magnetic field, temperature, electric field, in a way that that you know more or less how. For example, for the magnetic field, you know that there's the Siemens interaction, so that like with magnetic field, there is a Siemens shift delta in shifting of the resonance, so that if you as an experimentalist can know how to measure detunings from known resonances, and that's something that engineers and physicists have known how to measure for like many decades, then you obtain information on this delta, and hence you obtain information on the quantity that you want to measure. It's really just a signal processing problem. The question is like, how can you then measure those detunings from resonance? Okay, but all of quantum sensing works because there is a certain energy difference that shifts as a function of a certain parameter that you want to measure. Okay, this point doesn't get made a lot, but I think it's extremely, extremely important. But there's a catch. The catch is that this quantum enhanced way of measuring things only works while the spin is well described by the laws of quantum mechanics. Everything that starts quantum dies classical. This is the reason why we live in a classical world. When quantum objects start interacting with each other in uncontrolled ways, we lose, if you have heard this term before, we lose coherences, we lose the order of the quantum interactions, right? So that after a certain time, the spin is no longer well described by the laws of quantum mechanics. The spin is just a tiny little classical magnet. And from that point on, you can no longer obtain this quantum enhanced information from the spin, right? And it turns out that yes.
Sorry, just to clarify, so the 2 microseconds is after the onset of the magnetic field?
It's after the onset of you preparing, of you starting to do the sensing procedure. So the 2 microseconds is after you initialize your spin in a certain way and start manipulating it. That's correct then. It's 2 microseconds from the onset of the magnetic field that you're measuring, right? And in crappily naturally occurring diamonds, this time is about, if you have heard this before, this is T2 star. in crappy diamonds is about 2 microseconds at room temperature and in bulk. This is absurdly long. If you use better materials, you can even increase that long, increase this time. And this characteristic time is sometimes referred to as coherence times, right? So the question is like, well, is there any way to be smart about it and actually try to control this spin somehow so that it behaves for longer and longer and longer than those 2 microseconds as a quantum object? Think of it as if you had a swing, right? You have a swing that is going back and forth. If you kick the swing at particular intervals, you can keep the oscillation going. So is there any way that we can kick this spin so that we keep it going as a quantum object for longer and longer? And it turns out that spins and quantum objects made out of matter can be controlled using electromagnetic fields. We can give kicks of light. This is basically what was discovered in the field of magnetic resonance. And by giving those kicks of light, you can actually keep the spin living longer as a quantum object. So if we think about it in terms of a signal processing problem, again, it's like a signal as a function of the time that you're observing the signal from the sensor. Like at some point, when your signal goes to 0, you're toast. You don't get quantum enhanced information out of the sensor. So the idea is to apply kicks so that you have the signal going for longer and longer, so that when you Fourier transform it, you can have a better spectrum detail, right? So in the next slide, which is going to be the most technical slide ever in this presentation, I'm going to show you like one way of giving those controlled light kicks onto this sensor, okay? But don't be afraid. This is the big picture. I don't want you to forget the big picture, which is having a tiny little sensor that you can bring in close proximity to a tiny little sample and measure the tiny magnetic fields produced by this sample. So here's my example of such a spin control experiment. So in my past, I measured magnetic fields using this spin by giving it kicks in the shape of a pulse sequence known as rotary echo. Rotary echo has been known since the 60s, almost in nuclear magnetic resonance, and it's depicted in nuclear magnetic resonance jargon to the right there. This is what this means. This means that I am applying to my diamond crystal a constant microwave pulse tuned to the original resonance between the 0 and 1 states, up and down. Okay, so the microwave is constant, is always on, but at particular intervals, I flip the phase of this microwave. So I'm sending a pulse like this, and like at certain kicks, I do a 180 phase jump. And then at some point, I jump again. And I do those phase jumps in those continuous wave microwave excitation. This is a pulse sequence, electromagnetic kicks, that was used in nuclear magnetic resonance to correct for driving field imperfections, to correct for imperfections in your microwave source. But it turns out that it's known not to correct for detunings from mismatches from the resonance. And if you remember from my last slide, mismatches or detunings delta from the resonance is exactly what we want to measure. So this makes this kicks good to do magnetometry. So this is what this looks like. So here you can see the signal that I collect from this spin, which is just fluorescence normalized as a function of the time that I apply those CW with phase jumps onto the diamond. Okay, and what you can see is, if you know what I'm talking about, this is just a Rabi experiment with some modulations. So for example, if the signal is at the top, say it's emitting at the maximum of fluorescence, the spin is in one state. If you are at the minimum of the fluorescence, the spin is in the other state, and everything in between signals that the spin at the moment that I measured it was in a superposition of 0 and 1. Okay. And we can actually do some complicated math called average Hamiltonian theory to actually determine how the signal depends on this delta that I want to measure. And now we can actually know how to measure delta using the signal. We can then Fourier transform, we can Fourier transform the signal and obtain what you can see to the right, which is some sort of spectrum. It doesn't matter, but all that you see is that you can actually see different peaks corresponding to different deltas, to different detunings delta that you want to measure. And it turns out that here with this data that I'm showing you, this defecting diamond is feeling 3 different magnetic fields, the smallest of which is about 1/10 of the magnetic field of the Earth. So this is how we use those things to measure magnetic fields. This is how this spin is used for sensing magnetic fields. Okay? But again, not important. The important part is this big picture, having a tiny sensor that can sense tiny little magnetic fields in very close proximity to a sample. So while I ask you what your questions are, I just want to make sure that I make it explicit that I've just shown you a quantum sensor that works at room temperature and in noisy environments. That is, in the noisy environment of the solid state lattice, in the noisy environment of the proximity to a biological sample. And while you think if you have questions, let me leave you with a question, because that's one of the questions that I get the most. I've just told you that this tiny sensor, right, exists in diamond at room temperature, and the resonance between the 2 spin states is about 3 gigahertz. If you look at thermal energies at room temperature in gigahertz, this corresponds to about 6,000 gigahertz. So is this a paradox that I'm actually working with a 2-level system with a quantum sensor that has an energy difference of 3 gigahertz and that's immersed in a 6,000 gigahertz thermal bath? So what are your questions? And does anyone want to volunteer an answer to this question of whether this is a paradox or not?
So presumably that discrepancy is accounting for the shortness of the coherence time, right?
Yes, exactly, right? So the thing is there going to be thermalization? Yes. Does it occur immediately? No. There's about a couple of microseconds before thermalization. And again, after thermalization, there is no whatsoever quantum enhanced information that I can get from this sensor. The idea is that all the important information that I get from this sensor occurs before those thermalizations, right? That happens super fast, but it's not fast enough in the sense that we can still obtain useful quantum information before that. So it's not a paradox.
But you have to drive the system away from equilibrium by your radio frequency signal.
That's correct. So 2 microseconds is the free decoherence
relaxation.
Time.
Yeah.
Right. If I have RF kits, it's going to improve a lot. But in any case, it will never last forever, right? That's why we don't have fully fledged working quantum computers yet. So there's a finite time during which I can obtain quantum enhanced information out of this defect, and this corresponds to the time that it takes for this whole shebang to thermalize. So there's no problem that this thing is like a 3 gigahertz 2-level system immersed into this very, very fast path, because all the action that I'm concerned with happens before thermalization.
So Clarice, you're thinking of this as a very small sensor, but you need your magnetic field and you need radio frequency pulsing. So is that at an intensity where you can cope with the smallness of what you're trying to do? Because in an NMR setup, you have huge magnets, right?
No. So as I was saying, for example, it's with that crappy signal that I showed you there, we were already measuring magnetic fields on the order of 1/10 of the magnetic field of the Earth. The magnetic field of the Earth is about 50 microtesla, which is very, very tiny, which is way tinier than the magnetic field that you sense when you put your cell phone close to your face. So basically, those things can sense tiny magnetic fields, but they need to be close to the magnetic field source. So this is a proximity sensor. This said, there are two places that I know of, and many more under being researched, but there's like places at which this is already being put to use for practical applications. For example, Schlumberger, the oil company, they are already prospecting for oil based on signals from nitrogen vacancy centers, right? What people are doing now, they take so application number one. Application number two is thermometry in cells. People can make nanodiamonds, which are like 10 to 20 nanometer crystals of diamond containing only one of such defects. They shoot them into cells, and then via measuring a shift in resonance in the NV center, they can measure temperature changes inside cells. So those things are already sort of happening.
So what kind of temperature changes can you measure with a nanocrystal?
That I don't know. I don't know. If I had to give a guess, I would say it's sub single digit Kelvin, but I am not sure. To cut a long story short, I mean, at least for magnetic fields, we are talking here about like tiny deltas of magnetic field, right? And I believe if it weren't like sub-Kelvin deltas in temperature, people wouldn't be using this to track temperature in cells.
Right, because the normal assumption is that, I mean, that it's isothermal, and
exactly.
It's already very contentious as to whether there are real temperature differences and on what time scale.
I mean, one thing about temperatures is temperature is really a macroscopic quantity. So what you really if you have a single spin, you're probably inside the range of fluctuations. So what you're probably measuring is some statistical property, which may be temperature or not. Do you know what I mean? It's like the local, you're like measuring the local fluctuations inside the system.
So I don't know. The only other thing I want to mention about this inside cells is that, so remember, we need some control. We need lasers, we need microwaves, right? And apparently I'm told that the cells survive. Even if you microwave, you shine microwaves through, you shine lasers through. So apparently the cells are still alive, which I find quite cool, actually. But in any case, this is like real technology with some uses. So you are using this quantum sensor that happens at room temperature and in disordered environments, right? And it's working. So there's nothing paradoxical or unreal about this sensor. If you let me now, I really need to talk to you about what I really want to talk to you about. So up to like a couple of years ago, when I thought about sensors, I thought about the sensor in diamond and those type of sensors that you see there in the slide. But at some point, I sort of realized that many times nature overperformed nature-made sensors overperformed humankind-made sensors in crazy, crazy ways. In my past, I've worked with a couple of those, but here I would like to talk to you a natural magnetic sensor that I want to convince you that is quantum-based and that actually it bears a lot of resemblance to the spin in diamond. So this is really, really what I want to talk to you about. And in order to talk to you about sensing, biological sensing at the nanoscale, I really need to start talking to you about biology at the nanoscale, namely the chemistry. It's actually been known for many decades in a field called radical chemistry that magnetic fields can alter the final products of a class of chemical reactions that depend on spins. Here's how this is known to work in basic chemistry for things in solution, right? There's like a chemical reaction that's happening. At some point, the chemical reaction comes to an injunction. And at this injunction in time, depending on the spin state of a certain electron, if the spin is, and I'm going to use jargon, if the spin is measured by the environment to be up, the chemical reaction continues through one branch. If the spin is measured, that particular spin is measured by the environment to be down, the chemical reaction continues through another branch. And importantly, the macroscopic final products of those two branches are different. Magnetic fields, they can actually alter the spin states of the electrons. So a very fast interaction at some point in a chemical reaction with a magnetic field can actually macroscopically alter the final product of the chemical reaction and at much, much longer time scales because things happen like downstream. Okay, so this is known to happen at room temperature, in solution, in the gas phase, in the solid state, and for magnetic fields as weak as the magnetic field of the Earth, which again is smaller than the magnetic field that you sense, the magnetic field that you sense if you put your cell phone close to your face. And for reasons that I won't have time to explain fully, but hopefully I'll show you my simulations that corroborate that. The magnetic fields that are shown to actually be able to alter those spin-dependent chemical reactions are relatively small, okay, on the order of microtesla to millitesla. This means, without explaining too much, I can explain if you have questions. If you put those spin-sensitive chemical reactions inside a magnetic resonance machine, and you change a little bit of this huge field, it's not going to care. So actually, the magnetic fields that influence this sort of spin-dependent chemical reactions, those are small, very, very small, from microtesla to millitesla. So, and this is where the biology came in. And in order to talk about the biology, I really need to start talking to you about birds. Now, with all due respect, I really don't care about birds, but historically, everything started with birds. So I really need to talk to you about birds. So again, it's been known since the 60s or 70s that birds, when they migrate, they use, at least as a partial cue, the magnetic field of the Earth to migrate, to localize themselves. This is known without a doubt. And people were trying like different mechanisms to explain that. And at some point, the end of the 70s, some brave theoretical biophysicists, which was crazy for the time, they posited the following hypothesis, right? Which was crazy at the time. Well, were this type of spin-dependent chemical reaction happening under physiological conditions at some point inside the bird, birds and organisms in general, maybe they could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from these spin-dependent chemical reactions. Again, this was an absolute outrageous hypothesis, but people actually started looking, Oh, and the idea was, and this is not proven nor disproven in birds yet, but the idea was, for example, imagine that there were like a protein in the bird's eyes, that maybe it depending on the magnetic field, it sort of modulated the sensitivity of the bird to like light, to the bird's eye for light. In this sense, if this were true, a bird moving its head left and right, maybe it could see modulations of light intensity, which could give rise to like really seeing magnetic field lines. Again, this is not proven, this was a hypothesis, but people did start looking for proteins that could actually sustain those spin-dependent chemical reactions. And at that point, the only animal, because birds, the only animal protein that was known to be able to sustain these spin-sensitive chemical reactions was a fluorescent protein. It has a flavin that makes it fluorescent, a fluorescent protein called cryptochrome. Now, cryptochrome exists in the retina of birds, in our eyes, in the antennas of migrating butterflies, but it also, and I've checked and rechecked this sentence with bio people. All our cells have cryptochrome, because cryptochrome is also responsible for circadian rhythm regulation, okay, in our body. And it turns out that cryptochrome is, first of all, really widespread across the tree of life. And there have been many magnetosensing studies that macroscopically have implicated cryptochrome in one way or the other. So all those organisms there, from algae to like birds and locusts and mice and ourselves, we all express cryptochrome, and boxed in red are the organisms to which there have been some, again, macroscopic studies that linked somehow magnetic field sensing with cryptochrome. And it turns out that I am very convinced that evidence for cryptochrome-based magnetosensing is very, very widespread, but unfortunately at very disconnected length scales. So to the left of my slide, you have like the very tiny chemical length scales, and to the right of my slide, you have the next length scale step, which is unfortunately experiments with like whole birds, whole flies. Let me explain to you those two sides of my slide. So at the very small side, right, there is no whatsoever doubt that cryptochrome in solution works or interacts with a magnetic field in pretty much the same way that the spinning diamond does. Okay? And that's, let me tell you some of the very important analogies. For example, researchers put cryptochrome in a solution, like pure cryptochrome, and excited it with a laser. It has a flavin that absorbs light, gets excited, and then it excites emitting fluorescence. And they tracked, and that's the plot to the left top, the fluorescence of cryptochrome as a function of the time during which you're applying this excitation laser. Well, the first thing that you see is that this fluorescence is going down. You're killing the chromophore, right? It's called bleaching. It's nothing more than that. You're just destroying your sample. But when the researchers turned on and off a tiny magnetic field, and this is not an artifact, it has been tried with different magnetic field frequencies and intensities. As the researchers pulse a tiny magnetic field enough, what you can see in the inset there is that there's a tiny following of this fluorescence intensity that follows the magnetic field profile. Okay, this is an exact analogy to the spinning diamond. Remember, just by looking at how strongly the that blob in diamond was fluorescing, you could actually infer if the spin was up or down. Here, just by looking at how strongly the cryptochrome is emitting light, you could actually know if the chemical reaction continued through one pathway or the other pathway. And also, it's not super good evidence, but there's decent enough evidence to show that a cryptochrome at room temperature might have coherence times might be quantum for about 1 microsecond, which is also unusually long. Now, this is the chemistry, right? But we go from the chemistry to birds. For example, during migration season, those experiments are super hard, but again, it's like sample size of 21 every year, right? You put a bird inside a big cage during migration season, and then you mess up with the magnetic field around, and you measure where the bird wants to go out. You can do little experiments by applying RF in a way that messes up the spin mechanism, and the birds lose their orientation. Flies do not migrate, but I have no idea how that's done, but it's crazy. People can actually train flies to find food based on the presence of a magnetic field. I don't know how that's done, but that's done. And then people actually remove the cryptochrome gene from the flies, and then the flies lose the ability of finding food in the presence of a magnetic field. In a further experiment, the same researchers put back human cryptochrome in those knocked out flies, and the flies are back to being able to find food based on the presence of a magnetic field. Most of those huge scale behavioral experiments, most of the evidence is consistent with what's predicted by the spin model, but it's really very hard to make people talk, right? Because there's a lot going on between a molecule and a whole organism. So that's where we enter. The idea that we have started to do here is to actually bridge those two length scales. What we want to do is try to look, say, at single cells first, and try to observe at the same time the spin dynamics via fluorescence and like consequences downstream that might be affected by this spin dynamics. And in order to do this, what we're building are glorified microscopes with coils. The coils are there to mess up with the magnetic field. And this is not our microscope. Our microscopes are still being built. This is the microscope that I worked with at Stanford, where I was a postdoc. But the idea is that we would be the first to have a look, like, first of all, at how spin physics that you can measure, that it's there and we're preparing it correctly, how it affects downstream processes as measured by usual fluorescence markers. And when I talk that we're building microscopes, it doesn't look like the left picture there. What we have, again, this is not ours yet, but it's going to look very much like this. It's a big optical table with mirrors and electronics around. It's like an atomic physics experiment that works on a biological sample. And this is really the bottleneck of this flavor of quantum biology, which is like the influence of spin physics in biology. So what we're doing is sort of hijacking the same type of technology that is used to study and control, say, quantum sensors in diamond, and applying the same technology directly onto those living quantum sensors, because that's what they are. Those spins inside proteins, they are bona fide quantum sensors, right? So I believe that there have been many, many beautiful experiments in spin quantum biology performed by chemists, by biologists, and now I think it's time for quantum instrumentalists to push this field forward by providing us, like with high-tech instrumentation, to actually do quantum measurements in biological tissues, right? So this is what we're starting to do. I started at UCLA like in November of 2019, which is like 3 months before all hell broke loose. It's not a very good time to start a group, especially if you're in the business of building instruments. So we're very, very much delayed, but that's what we're moving towards. And we're still building 2 optical microscopes, a probe microscope, and we want also to have an electrophysiology microscope with coils. So the interesting thing is that differently from many biophysics experiments, we sort of change the order of things around. So in some biophysics experiments, you get the data and then you model it. In our case, from the start, we take simplified mathematical models, we make predictions, and then we want to see whether or not our models are good. And actually, you can use even, I mean, I have a theory friend who does things with like 20 spins and all, but we can do very simple simulations with like 3 little spins using basically Schrödinger, the numerical evolution of Schrödinger's equation in the presence of noise, basically the tools of hardcore quantum mechanics, plus some parameters gotten from the photophysics of cryptochrome, and we can start making predictions. And let me show you some predictions, right? For example, and we're not the first people to make those predictions, but our predictions agree with those. So I would like you to look at the top right plot. Okay, here's a theoretical prediction that that shows the magnetosensitivity of cryptochrome, that is how sensitive cryptochrome is to magnetic fields, as a function of magnetic field strength for cryptochrome molecules tumbling in solution. The first thing that you see, and I'm dying to take this curve experimentally, the first thing that you see is that this magnetosensitivity is not monotonic, it goes up and down. So at about 10 times the magnetic field of the Earth, at about 500 microtesla, the magnetosensitivity has gone down a lot. That is, as I said, for reasons that have to do with like spin dynamics, going to higher magnetic fields doesn't help. This is eminently a small intensity of fields phenomenon, right? That's one thing. The second thing is that, whoops, there it goes. The second cool thing in this plot is that it's not monotonic, and the peak of this plot is more or less at the strength of the magnetic field of the Earth, which might be a coincidence, or it might be that there has been some evolutionary pressure for the proteins to evolve, to work better under the conditions where it is. We cannot tell apart if it's a coincidence or not, but we think that people who do something called directed evolution of proteins could help us tell apart if there can be evolutionary pressures on where those proteins work well as quantum magnetometers or not. But again, now I can stop talking about birds, because this is way more important than birds. Historically, cryptochrome and birds have been studied, and cryptochrome remains the most well-studied protein that can sustain spin-dependent chemical reactions. But now there's more and more evidence at the macro level that many other proteins might be as spin-sensitive as cryptochrome, and those proteins are involved in super, super important, ubiquitous, like biological phenomena. Turns out that the production and regulation of reactive oxygen species is dependent on magnetic fields in a way that is consistent with the spin model. Glycolysis rates in cells, cellular acidification rates, stem cell growth in planaria, yield of DNA repair. Those are all things that depend on magnetic fields, on weak magnetic fields, in a way that really looks like there is an underlying spin phenomenon. Cellular autofluorescence, possibly from flavoproteins in mitochondria, is magnetic field sensitive, again, consistent with the spin model. And I would like to take a couple of minutes to explain this bottom picture there, because when I explain those pictures, people go, woo. So I have a friend who's a bona fide precision measurement guy. So what he does for a living is he builds hypomagnetic chambers. His name is Peter Fürninger. He's in Munich. And he builds very, very good Faraday cages, those hypomagnetic chambers. And then what he does with them is he puts cold atoms experiments inside, and then he measures like the variations of fundamental constants and stuff. So basically, his life is building those hypomagnetic chambers that have a DC noise level of about a nanotesla. Again, the magnetic field of the Earth is about 50 microtesla. So the noise level inside is way, way smaller. he started to hear those things about, oh, magnetic fields in biology. So here's what he did. He grew tadpoles, frog embryos, inside those hypomagnetic chambers for 2 days, because that was as long as he could do without asking for a biopermit, under 2 conditions. In one condition, he applied inside the chamber a mimicked tiny magnetic field on the order of the Earth. And in the second time, he didn't apply anything, just the nano Tesla. What he saw was that the frogs, the frog embryos, the tadpoles grown under the mimicked magnetic field of the Earth, macroscopically were okay. Whereas the tadpoles grown under just the nanotesla level, 30% of them, as you can see in this plate, they were macroscopically deformed. Other 2 groups, another physics group and a group in biology at Tufts, they have reproduced this result. I don't think it's published yet. And again, it's a macroscopic result, but given the fact that you're not even applying magnetic fields, but just taking away a tiny magnetic field, and you're getting such a strong result, everyone's best guess is that this involves spins, right? Which gives, if you melanin has a lot of rich spin physics, and embryo tadpoles, I believe, have melanin, right? That's, I think, gives its black color. So if I needed to bet, I would bet that it's something having to do with melanin. Another thing, research from the Army that also did epigenetic experiments in the absence of, like in a Faraday cage, also saw differences in methylation rates, so epigenetic changes. So all those things, none of those have been observed at the nanoscale, right? But I think that this type of experiments opens up like this huge Pandora box, not only for biology on Earth, but like, you know, like Mars. What's the magnetic field on Mars? Can we grow lettuce in Mars? Can we reproduce in Mars? And I think there's all sorts of like ramifications of this that people in many different fields should be thinking about, right? And this is actually what we're set out to do. We want to either establish or refute with like error bounds whether quantum mechanics can alter in a big way physiology, and can it be, and that's the important part, manipulated to technological and therapeutic advantage, right? Technologically, it's like, I'm not saying let's replace Google's beautiful quantum computer with a molecule, but if nature is using quantum mechanics to function, it knows darn well how to deal with noise, right? Which is the reason why quantum computers are not permanently working right now today. So can we learn some trick used by nature on how to deal with noise and apply this trick on top of existing quantum computing architectures? For example, can we build better quantum sensors using the laws of quantum sensing from nature? Or can we deterministically manipulate those endogenous quantum mechanical degrees of freedom in order to drive physiological outcomes, in order to drive those spin-dependent chemical reactions, which apparently are much, much, much more widespread than currently investigated, right? Imagine that, like, it's not going to happen today, not tomorrow, but maybe in 50 years if we start doing research now. Can we have, like, you know, an index? Well, if you want to change a certain reaction in a certain way, this is the magnetic field strength, direction, and frequency that you need to apply in order to drive those chemical reactions towards like disease mitigation or whatever it is that you're interested in, right? We're not the only people thinking about like very high-tech ways of studying those endogenous quantum knobs in biology. Again, you don't need markers. It's like optogenetics without markers. Everything that you need is already there. It's like learning how to tap onto those, how to control those to like therapeutic advantage. So there are many other people applying very high-tech to tackle different biological processes that involve quantum knobs. So this all I wanted to talk. I have to thank profusely my group who started a group, like who took on the challenge of being part of a new group in the middle of a pandemic in a theme, in a field that is many times that still goes misunderstood, right? I am always looking forward to talking to people, because I think that interdisciplinarity is really what's going to drive science forward in this century. And I like to finish my talks by saying, may the quantum be with you.
Thank you so much, very much, Clarice, for a fantastic, passionate talk. It was fantastic. Can I start with a question which is going off maybe a bit orthogonally? Your topic?
Yes, always.
In preparing for your talk today, I found a paper, I forgot the authors now. They look at the possibility of optical quantum computing in the brain, you know, where they, maybe you know the paper, I forgot the authors now, where they look at the possibility of having optical beams propagating along the neurons, you know, and to having entanglement across the brain. Have you looked at that direction of work?
It's Christophe Simon in Canada. He's very good. He's a theorist, and he's also like a quantum physicist by training, right? And I think, and that's part of my spiel too, right? I think we need quantum experiments to probe all those things. I think that, again, when I say those things, some people call it a heresy, but I really think that quantum biology is where quantum computing was like 30 years ago. There was a lot of theory, a lot of things like, oh, how can we put those things to work as a quantum computer? And then at some point, some pioneer people started doing simple experiments of like quantum gates. And now we have like this booming quantum industry. I think, and I like to think, that quantum biology is sort of in the same point, where we have a lot of theory that needs to be tested by high quality quantum inspired experiments. I think there are so many ways where nature could be using quantum mechanical degrees of freedom to function, but we will only be able to prove or refute once we have those new experiments. Right now, there's very little instrumentation that can actually unambiguously test for that, right? But I find it fascinating too, right? It's a very cool model that seems interesting to look at.
If it was true, it would be amazing, because it'll open up a new field of quantum computing. In like the brain is like a wet type of, it's all in water essentially. It's
right, but here again is the question, like, for how long does the things need to be quantum so that it can have an effect, right? And maybe we don't need super long, we just need the thing to be quantum for long enough to drive X or Y process.
So Clarice, can I just pick up on that point? Because when you were talking about your I mean, this was a lovely talk. I really, it's very inspiring and stimulating. We were talking about your diamond sensor. You made the point that the decoherence time is 2 microseconds, and if you want to acquire information from this, you have to apply your radio frequency pulses. You're taking the system and creating a non-equilibrium state in order to do this. And then when you come to the bird and cryptochrome, what's the equivalent of that? How does the bird maintain decoherence of a system which presumably also has I mean, I believe that the flavins in cryptochrome might have the equivalent of a spin system in it, but it will have a very short decoherence time. So how does the bird cope with this problem?
So you're absolutely right that I don't expect there to be any kicks in nature to extend coherence times at all. So let's think about what can be done within the coherence time, right? So one back of the envelope calculation of like how small a field you can sense is like, so spins, when they are immersed in a magnetic field, they precess, if you have a magnetic field here, they precess around this magnetic field with a characteristic frequency called the Larmor frequency. The Larmor frequency for an electron under the magnetic field of the Earth is about 1.4 megahertz. The back of the envelope calculation is that if you can do one full rotation before decohering, you can sense this. This is just order of magnitude, okay? So 1 over this 1.4 megahertz is about 700 nanoseconds. This means that cryptochrome would need to be at least coherent for 700 nanoseconds in order to sense the magnetic field of the Earth. And again, there is not complete, but some evidence that shows that it could be up to 1 microsecond. So that's good. The question is like, even so, this is a very small magnetic field, but proteins that could sustain those spin-dependent chemical reactions with shorter coherence times, they could still be sensitive to larger magnetic fields. If you're coherent for longer, you can sense smaller magnetic fields. If you're coherent for shorter, you're still a sensor, but you're a crappy sensor. You can sense only big magnetic fields. So the thing is, like, cryptochrome might be able to sense the magnetic field of the Earth, but what other magnetic fields might be happening inside cells and that might be shifting the equilibrium of those spin-dependent chemical reactions? For example, I have a hypothesis, and again, this is not based on data at all. I'm just very curious, and I think it might be possible. Ion fluxes that control everything inside cells are transient ion fluxes creating magnetic fields that are strong enough for a certain time to shift the equilibrium of spin-dependent chemical reactions. And if those proteins involved are coherent for shorter, are those magnetic fields large enough, right? Because I don't think, like, from a very tiny distance, I think even transient ion fluxes could produce like decent magnetic fields that could be sensed even by crappy quantum sensors, right? So it's nature is by no means using control pulses. I don't think so. I don't know. Maybe I'm wrong. It's awesome if I were wrong, but I don't think it is. But I still think there's a lot to be sensed and interacted with, even for crappy nanosensors. Cryptochrome, if you will, it attracted attention because it's very unusual, right? In the same way that the NV Center is very unusual. But I don't believe you need to be that unusual, because maybe what you need to sense are larger transient magnetic fields.
So, I mean, biology's always been known to be strongly electrical, as you pointed out. You know, every cell has a resting membrane potential, and it only maintains that by using an enormous amount of energy to push ions back and forth across the membrane. So it's always been fundamentally, you know, sort of electrical. So to my mind, that's always raised the possibility that therefore it is also sensitive to magnetic fields, because Maxwell's equations tell you that it should be. So in principle, would it be possible to account for some of these phenomena just through a purely classical effect rather than having to come down to a quantum level? I mean, we treat membrane electrophysiology in a perfectly classical way. Is there some argument, you know, back of the envelope calculation that says, well, actually, no, it's not adequate to account for what one sees?
Not that I know. Let me see how I'm going to. There are weird effects that would be hard to explain if you don't talk about spins. For example, when tiny little magnitudes of fields are involved, right?
Mm-hmm. So the flavin example would be a good one, right? I mean,
yes, and like when people do experiments, say with birds, right? The idea is that there's an electron that is interacting with the Larmor frequency of the Earth, and then again, the Larmor frequency is about 1.4 megahertz. If you put a tiny magnetic field AC that oscillates with like 1.4 megahertz. Again, the magnetic field of the Earth is like 50 microtesla. Researchers put onto the bird like 200 nanotesla of AC field of 1.4. If the spin model is correct, you're basically you're tapping onto this resonance, and you're making this guy go crazy, and the birds lost the orientation. This is crazy because it's a tiny effect. The other thing is like this absence of this tiny magnetic field, right? If this and that caused like this crazy tadpole stuff, I think that the electric field associated.
The tadpole experiment was very striking. I have to say that I hadn't heard about that, and that's really amazing.
Yeah. Right, and those, but again, I think that there might be things inside cells that depend on magnetic fields in a classical way. I'm all for that, and I think that there is a subset that's, to the best of my knowledge, right now can only be explained by spin.
So you would advise us to be wary of being in magnetic fields?
In small magnetic fields, right? I mean, it's crazy, right? We don't know what those things do.
We're immersed in them all the time.
Right? I mean, it's crazy. I think those things need to be systematically studied, and like so to a physicist, I would describe what I do as like light matter interaction, where matter is biomatter, right? And the idea is that electromagnetics in biology is like taboo in biohydrogen. It's like taboo in the West. People in the East, in Japan, Japan has one of the strongest spin communities in the world. Like in China, in Japan, in Russia, people have been doing electromagnetics in biology experiments for decades. If I go to like compilation of articles of electromagnetics in biology, it's all like Russian papers, and there's very little systematics on it. In one paper, people are applying 1 gigahertz at like 50 microtesla, and the other one like DC. It's a mess systematically. But there's like a lot of funky stuff that is going on that would merit, like it's become taboo in the US at least, which is a pity, because I think it could be like a very nice knob to do pharmaceutics, you know, another way of changing things that is not like chemically changing things, it's changing things electromagnetically.
Can I ask a bit a crazy question? You know, there's a lot of people putting up resistance against cell phone towers, yeah, and they say that the magnetic field has some influence and so on. What do you think about that? I mean, in principle, with your equipment, you should be able at some point to measure if these protests are well-founded or not.
So here's the thing, Gerhard, like, I may be wrong, but when I think about like towers, I think about very strong magnetic fields. The thing is that those chemical reactions, they occur at relatively low fields. So I don't fully understand, like I'm sure that people feel the presence of those power lines and stuff. I'm not sure they have to do with like spin physics, because for that, we would need lower fields in general, right? I don't know if you have, those things like the Havana syndrome where people
yes.
So I wonder if someone has investigated whether it's not like an attack with like a microwave of a certain frequency direction that is not strong. It doesn't need to be strong in principle, right? That's the advantage of this, of tapping onto this mechanism. You don't need like a super strong gun or anything. So I wonder. I think there are many things that could be investigated on this field.
Yasuhide, do you have questions from the
yes, just naive questions. For example, Chrome has many atoms, nucleus, electrons. Which spin are you measuring?
Yes, I cheated because actually it's not one spin, it's two spins. In cryptochrome, there is a chemical reaction that is spin sensitive to the state of two spins. When two spins interact, it's two radical spins. They're called a radical pair. And the idea is that there is one spin that is from the flavin, and the other spin is thought to be coming from a tryptophan.
Which one? Sorry, I didn't.
Tryptophan. Tryptophan. I think it's tryptophan that is pronounced. So the flavin, the pigment, and tryptophan, which is written, I think, like the tryptophan.
Okay. I'm not familiar with this word. Okay, please go ahead.
So let me see how Wikipedia describes tryptophan to me. It's like an amino acid. No, it's an amino acid. It's an amino acid. What I know about tryptophan, which makes it very interesting, is that apparently it's not super common. It's not super abundant, which I then wonder, like, something that should I be looking for more things that have tryptophan?
It's a kind of amino acid.
Yes.
Okay. Yes. Then the spin comes from the nucleus or electrons.
Electrons.
Okay. Yes. It makes sense. Thanks. Yeah.
A nuclear spin is a very poor quantum sensor. It's a very good memory unit because it doesn't decohere very fast, but it's a very bad quantum sensor because it doesn't talk to the environment a lot. So this is why quantum sensors in general are electron spins.
Okay, thank you.
Davide, do you have questions? Thank you, Yasuhide. Davide, do you have questions or?
Hi, Davide. Nice to meet you.
Hi, Clarice. Nice to meet you as well.
Davide works for
Zeiss.
Oh, nice.
I think you know the optical.
Oh, yes, of course. Nice.
Yes.
Very cool.
So great presentation, extremely interesting. Sorry I wasn't able to join right from the beginning. But can you hear me? Yeah.
Yes. Yes.
So I muted correctly. Yeah. So great presentation, very interesting. No specific question, I'd say, from me at the moment, which was simply great, right, to hear this perspective, right, on the impact of magnetism and also on the potential that lies, right, in magnetism, also from a sensing perspective. So as you probably can imagine, our research equipment is in some way, right, based on the sensing concept, although it's mostly optical. We have also integrated types of equipment, and one of the areas we're interested in. But I don't have that deep technology perspective. I'm more on the business and strategy side. But extremely interesting to hear your work and your also passion for it.
So some of the things that I'm working on is to try to sell the idea. I mean, it's all the same thing, actually, of like a microscope with a spin contrast, right? So the idea is that the contrast in your microscope is this different in fluorescence given by different spins at different magnetic fields.
Yeah.
So basically, our instrumentation is going to be like a spin microscope, a microscope that uses spins as a contrast mechanism, actually, right?
Yes,
so
is an additional, essentially like the fluorescence concept, but an additional dimension based on which you can identify and highlight specific details or specific parts of the image.
The idea is that we would use, as we're using, like the autofluorescence of proteins, and this autofluorescence in many different proteins are spin dependent, right? It's magnetic field dependent. But the thing is that protein A depends on magnetic fields differently from protein B than for protein C.
Yeah.
Right. So the idea is that if you have this index where you say, well, I get the maximum fluorescence for protein A at like this B, this magnetic field, then for the other protein at this other magnetic field, and for this protein at another magnetic field. Again, not now, but maybe in 50 years, maybe you could like.
You can locate the different proteins the same way as you can use, let's say, spectroscopy to identify different types of, different elements or different chemical compounds.
Exactly, right.
It's really interesting.
And yeah, again, it's just an idea. It's not a technology. It's again, those things are going to take time, right? But if nobody does the research right now, it's never going to happen. But it's
yeah, but maybe we will have some opportunity to stay in touch and work on some of these aspects. But again, I'm not on the research and development side, but I'm certainly taking note and keeping that in mind.
Oh, exciting!
Yeah.
Thanks for coming.
Yeah, absolutely. Sorry for not being on video. It's a bit late here in Europe, and I was able to join only after the beginning. So I just
no problem. Oh my God! Yes, it is 10 midnight there. Oh my God.
Yeah, it's there was actually some confusion because we just had we just changed the times, so it's actually 1:30. Yeah, but it's
oh my God, go to sleep.
It's a good time because I'm sure that I can make it, right? But it's just I'm
oh my
at
God,
the end of the day.
Super bad.
To prepare for this, I heard your talk, Clarice, to the Cambridge Society, and I think it was 3 a.m. for you, I think. So,
yes.
I'm with organizing the Trinity in Japan, I'm quite egoistic, so I always have it during daytimes in Tokyo about
cool. But isn't it cool now that we can Zoom with people from all over the world? It's super cool, I think.
Oh, it's amazing. I mean, this without the virus, I think I wouldn't have started this Zoom. Even Zoom wouldn't exist probably.
Yes, right. It's crazy. It's really crazy.
So I think if we have all asked you a lot of questions, I think we have lots more questions. So maybe some of them, if you come on, it's on 2nd of June, 2022. Yes. You have to take care because of the time zones. It might be 1st of June for you. So you have to take care of the timing.
Yes. My Google Calendar told me it was on the 1st. Yes.
Yeah, it depends on the time zone. Here in Tokyo, it's 2nd of June. For Jeremy Gunavardena, who you just talked to, So I think maybe he will collaborate in the future. Who knows? I mean, awesome. You're working in very, very close areas. I think. And Jeremy, I know for ages because he was elected on the same day as a fellow of Trinity, research fellow of Trinity, long time ago.
I still envy you because you can go through the grass. I could never walk through the grass because I was just an MPhil student. I was not a fellow because you could cross the grass.
That's how they say, it's kind of curiosity, but that's life in Trinity. Yes, and we have to do that, of course, especially it's fun in front of all the tourists. And do you know what happens if somebody who is not a fellow walks across the grass?
No.
The the potters will wait at the edge of the grass because the potters are not allowed to walk across the grass. So the potter will wait at the edge of the grass, and then he will challenge his tourists and say, "Are you a fellow of this college?"
So if you go back, are you still allowed to go to the grass?
No, I'm not a fellow now. I'm a past fellow, but not I'm a fellow.
Oh, I see.
But that's not, it's not such a thing. No?
Cool. Anyway.
Okay.
But
yeah.
Thank you. Now I know that you guys are here, and now I'll start joining more.
Oh, I forgot if I put you on the mailing list, but I'll put you on
Yes,
the mailing
I am.
And please join all our events. And once the traveling becomes more easy again, if you come to Japan, and you told me there's a big
I'll let you know.
activity here. So if you come
in
here,
quantum bio,
we can even make a Trinity focused event here, you know, and
cool.
We can organize that. I have some places where we can get people together. And
awesome.
We can organize a Trinity event.
Awesome.
You know, I want to promote that, to use that as a platform.
And for the other thing about the quantum computing stuff, if I can help anyhow, let me know. I know a lot of people. I'm trained like in the field of quantum computing, so
sure.
If I can help. Yeah.
That's the Boltzmann branch, you know. I used to have
your other hat. Your other hat.
I have many hats. many hats. I'm at the moment, one of the projects is, you know, I have a company here. So one of the projects is building a regenerative medicine company. So I'm working with a lot of venture capital firms now, doing the fundraising for investment raising for that. Okay, thank you so much. And hope to see you very soon again on Zoom, I guess. And maybe at some point we can do an event here in Tokyo when you come here.
Thank you, everyone. Thanks for your patience. Thanks for having me.
Take care.